Thanks. I'm really excited for the opportunity to share with you all our new algorithm for accurate segmentation of cell imaging data. There's now a number of different available multiplex protein imaging platforms. The underlying technology for each of these is a little bit different, but the output is essentially the same, which is a high-dimensional image where each channel in that image corresponds to one of the proteins that is being acquired. This data is really useful because it allows us to look at the co-expression of these different proteins within the individual cells in the tissue that we've imaged. In an analogous way to how single-cell RNA sequencing or cytOF data might be analyzed, we can look at the patterns of co-expression of these protein markers in order to understand what the cell types are in this imaging data. However, the really exciting thing is we can take that a step further, and because we have the location of each of those cells in the original tissue that we image, we can map back those specific cell phenotypes or cell subtypes to the original sample. For example, we can see how cells of a certain subtype tend to cluster or are spatially located near those cells of that same type or cells of different type. This is really powerful and is a great lens to looking at either healthy or diseased samples across a range of different conditions. However, these types of analyses cannot be performed directly on the image data that comes off of the instrument. These images are essentially just matrices of pixels, where each pixel in the image corresponds to the expression value of the specific channel that you're looking at. In order to understand what's going on at the level of a cell, it's necessary to first define where each cell is located in the image. This process is known as cell segmentation, and in tissue, it turns out to be a fairly complex challenge and has been a major barrier to analyzing these imaging data sets. The reason this is challenging is because tissue data is quite complex. These cells are very densely packed next to one another. There's often a lot of heterogeneity in cell shape and cell size, even for cells that directly adjoin one another. And there's a large degree of variation across different imaging platforms, different tissue types, and even different experimental conditions. To address this issue, we created a novel deep learning architecture, which we're calling Mesmer. Mesmer is specifically designed for segmenting tissue imaging data. The inputs to the Mesmer model is a two-channel image. The first channel, which defines the membrane or cytoplasm of the cell, is used for cell shape. The second channel is a nuclear marker, which defines the nucleus of the cell. We pass these two channels to a normalization function and then directly into the deep learning model. What the deep learning model does is predict both the center and the shape of each cell and each nucleus in the image. We then take these predictions and post-process them to create the final segmentation output, which is a mask representing the shape of each cell and each nucleus in the image. As you may be aware, deep learning models require large amounts of training data in order to get accurate performance. We created TissueNet, which is the largest data set to date of cell segmentation, training data for cell segmentation. TissueNet contains more than 1 million paired annotations for both nuclear and whole cell data. If you're interested in the process that underwent the creation of TissueNet, you can check out our preprint, which I've listed here below. One of the most important considerations when constructing a training data set is that it has sufficient heterogeneity to represent the range of different examples you want models trained on that data set to handle well. To this end, we made sure to include images from a range of different tissue types so that models that are trained on TissueNet would work well across a range of different tissues. We used TissueNet to train the Mesmer model and then benchmarked it on a held-out portion of the data set. Shown here is the accuracy as assessed by the F1 score, where F1 score of 0 would indicate no agreement with the ground truth, and an F1 score of 1 indicates perfect agreement with the ground truth. What you can see here is that Mesmer is substantially more accurate than previously published cell segmentation approaches. To demonstrate this a bit more visually, we generated ground truth segmentations for colorectal carcinoma. What we've done is color each cell by the ratio of the size of the predicted cell to its matching ground truth cell. So for example, in this true segmentation data, everything is colored gray, corresponding to this 0 log2 full change between the predicted and true size, and the F1 score is 1. If we look at this same image and the segmentations generated by Mesmer, we see overall that many of the cells are colored this, middle gray area, indicating we're doing a good job capturing the size of these cells, but there are rare cells on the extreme ends of this log2 spectrum. When we compare that to the previously published models, you can see a dramatic difference with a large increase in the number of extreme colored cells on both ends of the spectrum, as well as cells that are completely missed by the methods, which is a major limitation if you're analyzing tissue imaging data. One of the benefits of TissueNet having such a heterogeneous collection of tissues and image platforms is that models trained on it are really generalizable. Shown here is example output from Mesmer on 4 different tissue types and 4 different imaging platforms. The important thing to note here is that this is the exact same version of the model with the exact same post-processing parameters and no manual tweaking required for it to work across this range of different tissues, which is currently a major downside, I would say, of many segmentation approaches, that they require a lot of fine-tuning. But because TissueNet is so heterogeneous, we can train a single model that works out of the box on this diverse set of data. We next wanted to evaluate how Mesmer compares to human annotators. We enlisted a number of pathologists to perform a blinded evaluation where we showed them 2 segmentations of the same image, one generated by the algorithm, one generated by humans, and asked them to evaluate in a blinded fashion which was more accurate. What you can see here is that these ratings, while there's some variability, are all clustered right around 0, indicating that they had no preference between the model's annotations and the human annotations. When we break this down by tissue type, we see some differences. So, for example, immune tissue, there was a slight preference for the model. The colon tissue, there was a slight preference for the annotators, but overall a similar pattern where these annotations are all clustered around this 0 axis, indicating that we've actually achieved human-level performance for this task, which is really exciting. Now that we have this accurate segmentation, I wanted to highlight some of the cool applications that we can use this for. As I mentioned before, Mesmer generates segmentation predictions not only for each cell, but each nucleus within each cell. What this allows us to do is look at the subcellular localization of imaging markers. So on the top here is a number of different proteins that have known subcellular localization patterns, and on the bottom is the corresponding segmentation predictions for both the nucleus and cell of each cell. What we can do is use this to compute the subcellular localization of each protein marker, and then do that same calculation for the ground truth data for both the nucleus and each cell. When we compare these to each other, we see that there's excellent agreement between the subcellular localization prediction of each of these proteins from the generated from Mesmer's segmentation, as well as from the ground truth segmentation. Another common task is quantifying cell subtypes. To evaluate how Mesmer impacts this task, we generated segmentation predictions from a data set including 3 phenotyping markers: CD8 for T cells, CD14 for monocytes, and keratin for tumor cells. We generated segmentations via Mesmer and also from our ground truth data from the annotators. We then used the same gating scheme to classify each cell into one of 4 potential subtypes. What you can see here is that in each image, the proportion of each cell in each cell subtype in the ground truth data agrees quite closely with the proportion of that cell from Mesmer, which is really important sanity check when using these methods to evaluate something complex, such as the different cell types that are present in the TME. Finally, we wanted to make this easy to access, so we've created a number of different ways for users to run the model on their own data. We have cloud-based approaches such as the deepcell.org website, plugins for ImageJ and QuPath, 2 popular analysis software, but we also have command line tools such as a Docker image and a Jupyter notebook so that a number of different users can access the model in sort of whatever way works best for them. All of the details are in the preprint, which I mentioned below. You can also check out the tutorials on our GitHubs. Finally, I wanted to thank everyone who was involved with this project. This was a really great collaboration with Dave's group at Caltech. Especially wanted to thank Geneva, my co-author on this study, both of my advisors, Mike and Christina, as well as Liat, who now has her own group at the Weitzman. Thank you for your time, and I'd be happy to take any questions.
No, there's one question here from Sweta Murali. How well does Mesmer perform in multi-layer tissues? For example, if there are cells partially obscured by cells from above.
Yeah, so I would say it's not superhuman. It was trained using human annotation. So if you, as a relative expert, can look at the tissue and get a sense for what a accurate segmentation should be, the model is going to get about that level of performance. For tissue types where it's just so crazy and even a human looking at it isn't really sure what to do, the model is not going to be able to sort of solve that problem for you.
Thanks.
Another question, does Mesmer work for 3D images, such as 3D confocal images?
Not yet. So if you have Z stacks of images, right now what you can do is run Mesmer sort of serially on each of those slices, and then you can stitch the predictions back together. In order for a model to understand how to do 3D segmentation, we would need 3D training data. And that, as you can imagine, is very laborious to create. So it's on the to-do list, but currently the model can be run on each of the 2D slices independently, and then you can stitch them together to get 3D segmentations.
Great. There's also a question here from Joanna Petrescu. It's very impressive how well your tool works on multiple tissue types. Have you tried it for segmenting cell types with more elaborate morphologies, such as neural tissues, neurons?
Yeah, so you may have noticed that there was no brain data in this TissueNet dataset, and that's because it really is such a fundamentally different problem. I think that's one area where 3D might actually be easier rather than harder. When you take a 2D slice of the brain, it's often really hard to figure out what axons are going to what neurons and how these things should be connected, because you're really cutting through what's sort of like a ball of spaghetti. And it's only if you had a more 3D structure that you would even be able to understand how to link those together. So right now in the brain, we're saying we can do nuclear segmentation, and anything more than that, I think, is going to be require more sophisticated approaches.
Thank you.
Do we have time for one more question? So there's one question from Dihu. How does Mesmer perform when the membrane signal is not fully clear? For example, in some live imaging data?
Yeah, that's a great question. And also, for example, what if you have cells that just don't express the membrane marker that you've imaged? This is a common problem. And everyone has been on the quest for the perfect membrane marker. I think we can now definitively say it doesn't exist. And so you just have to bake that into the algorithm. What the algorithm does is when it's unclear, exactly what the shape of the whole cell should be, based on that membrane marker, it will default to just doing nuclear segmentation, so that if you, for example, have an epithelial marker as your membrane marker, and there's immune cells that are in your image, it's not going to just skip them. It will just segment the nucleus in the cases where it's not clear what the shape of the whole cell should be. For those cells that do have robust, strong staining of a membrane marker, it will perform whole cell segmentation on those.
Awesome. Thank you very much, Noah.